Thank you very much. My name is Shigeyuki Kakizawa from Japan, and the title is Synthetic Defined Media for a Minimal Cell. Last year, we presented serum-free media for Syn3 and Syn3b, and we successfully removed serum from media, the SP4 media. So our serum-free media includes 3 or 5 types of lipids, and the 2 types of lipid carriers. And then we named the new media as a LPC, with which stands for lipid, PBA, and the cyclodextrin. And we made 2 types of the new serum-free media. And then Syn3b and Syn3 can grow well. And there is no visualized bond in the new LPC media. So the serum was removed, but some undefined component still remains. For example, the mycoplasma broth base, bactotrypton, fresh yeast extract, or yeastrate. So based on the serum-free LPC medium, we try to replace these components with the defined components. Here is a trial to make a fully synthetic media. Here is LPC9, a serum-free media. And then we remove the PPRO, trypton, fresh yeast extract, or yeastrate. And then instead, we added mixture of amino acid, vitamins, and the bases like this. This is a defined media. And this is just control. And as a result, in our defined media, the Syn3b cell can grow very well. So the mixture of amino acids, vitamins, and bases could replace the undefined components of SP4 media. Here is the first summary. So SP4 includes the serum, but our serum-free media does not increase the serum. But here we removed all undefined component except peptone, and we added mixture. So the first version of synthetic medium could be developed. Next, we tried removing peptone like this. So with peptone, and we tried to make a defined medium without peptone like this. And we also added mixture of amino acid, vitamins, and bases. As a result, growth was not observed in the absence of peptone. And then we tried various concentrations of peptone, but no peptone. In the no peptone, there is no growth observed without peptone, but growth occurred at 0.5 per liter peptone, really small amount of peptone. And then also slightly improved growth was observed at the higher concentration of peptone, like that. Then we tried to make a media with synthetic peptide. We ordered a lot of variation of synthetic peptide through the company. And we ordered the tri and the tetra peptide in total of 16. And we mixed them in one media. And also we ordered hexa and hepta peptide like that. And also nona and deca peptide like that. And we added the glutathione or not, and we try to cultivate Syn3b cell. As a result, here is a tri and tetra peptide, synthetic peptide. No growth was observed here. We don't know why, but in the hexa and hepta or nona and deca synthetic peptide, Syn3b can grow very well. So peptone could be replaced by the synthetic pure peptide. And here is a result of Syn3.0. And the growth is not well, a little bit, but really slow compared to the Sin3B. And here is a result of growth speed of Sin3B by plate reader. Here is red is SP4 FPS, and green is SP4 LPC9, our serum-free media. And then gray here is a defined media with peptone. And then we added, removed the peptone and replaced with a tri and tetra synthetic peptide. Almost no growth were observed. In the hex and peptide, really good growth were observed. And here is also. So Sin3B might prefer hexa and tetrahepta peptides. We don't know why Sin3B cannot grow in the tri and tetra synthetic peptide. And here is a result of Sin3. Almost same, but Sin3 showed slow growth in the defined medium, like this. And also in Sin3, tri and tetra peptide were unable to fully support the growth. Here is a summary. Based on the serum-free media, we successfully constructed a synthetic defined medium. And the mimer cell require a polymerized peptide as a nutrient source, in addition to the singular amino acid form. And this defined medium is expected to be applicable to various purposes, such as carbon-13 or tracer experiments, metabolomic analysis, and proteomic analysis, and so on. And we can control the amount of each component in the media. And I'm just happy to share the recipe to make the serum-free media or the synthetic defined media for Sin3. That's it. Thank you very much.
Shige, this is dazzling and important work and is going to save our community potentially a whole lot of money. I had always assumed that the mycoplasmas, because the oligopeptide transporter was essential, that mycoplasmas, there was at least one amino acid that couldn't be imported as a single amino acid. With your peptides, can you tell which amino acids have to be included in a peptide and which ones are not necessary? Do you have that knowledge yet?
Yes, actually, the most amino acids are included in our peptide, except the cysteine here. For example, the hexa and the hepta, the mix of the three synthetic peptide, only cysteine is absent. So maybe cysteine is incorporated in the singular amino acid form, but I have no idea about the other amino acids.
But we know the knowledge that cysteine wasn't, so could be brought in as a free amino acid. We had known that previously, because if you use the not tryptone, but or not peptone, but there's another product that gives you digested a protein digest that has no cysteine, that is anyway, we knew it wasn't cysteine, but you would think that you could start working with subsets of your peptides and figure this out, which ones could get free amino acids in and which one couldn't.
Actually, we added the all 20 amino acids in the singular form.
Mm-hmm.
So in addition to that, we added the synthetic peptide.
Mm-hmm.
So in the if we increase the amount of singular amino acid in the all of the 20 amino acid, Sin3B cannot grow without.
But
peptide.
what I was thinking is that you could use, as opposed to all of your so if you tried growing it on your like HFA, TKE, or different versions of your hexa and nana and deca peptides, you could figure out maybe which ones would support them by itself.
Yeah, that is interesting, but we didn't do that. So yeah, thank
Okay.
you for your advice. Yeah.
Zan and Bogomil have questions. Zan, you're first.
Yeah, that's just beautiful work, by the way. So I have two questions. There is already a known transporter that could do peptides. They weren't quite sure if it was only tetrapeptides or tripeptides. Do you think that there's another transporter for these longer peptides? And then my second question is, when you talk about the bases, are you including nucleosides as well as the nucleobases, or only the nucleobases?
The first question. Thank you for your question. The first question, I have no idea about that. So I don't have any information for the peptide, the transporter, importer. Mm-hmm. But maybe Syn3B has such kind of transporter. But
it does. It has the ability to at least break up the peptides. It requires ATP, but it can do it. So this is, going to be interesting in comparison to the growth rate. But what about the nucleobases and nucleosides?
Actually, I have a list of the mixture. I added like this: glutamine, uracil, thiamine, cysteine, adenine. We added 5 bases, mixture of all of 5 bases.
But it's just the bases, or did you put? It's a little small. I can't read it. Do you have also the nucleosides with it? Because we think the transporter can do either nucleosides or bases.
I added the only bases. So,
wow.
Okay.
That is fantastic.
Fabulous. So, Shige, do you have plans to do metabolomics with this work?
I'd like to do that, but not yet.
Okay.
Actually, we tested the carbon-13 labeled glucose, and then we tested to detect the protein, but there is no the signal in the protein from the glucose. So maybe next time.
Bogomil, you're up.
I can't hear you. Maybe muted.
Nothing, Bogomil.
Well, can I ask one more question? What's the fastest doubling time you observed?
Almost same. Actually, we didn't. Yeah, almost same. Actually, I didn't calculate the exact time of the doubling time, but the slope is almost similar, but a little bit slightly slow growth compared to the serum-free media or SP4.
Okay. Seems like you've got a longer lag phase.
Yeah.
Are you going from SP4 into your synthetic media, or are you starting all along in your synthetic media?
We first cultivate the media, the cell with SP4 first, and next day we washed and diluted and then input it into the new media. So
okay, all right.
Yeah.
Bert, Bogomil never could get the sound.
So this is related to the question on the oligopeptides. I think JCPI has an OPP system, and we've studied in the past an homolog, and that was actually very good in transporting non-americ peptides. So it's So there are basically OpenP homologs, but I think the one that is present in JCPI is actually geared towards transporting hexa, hepta, nona, deca peptides. So it's
Oh, okay.
Thank you very much.
Do you have a citation for that?
yeah, I will afterwards put it in the chat. Yeah.
Thank you.
It's a very
Thank
old
you,
paper, but it's still true.
Okay.
Can you hear me now? Sorry.
Go, Bogomil. It's your turn.
Sorry, I just had to log in somewhere else. Just quick question: was this ever tried in the solid media on plates? I am just interested if the cell behave similarly. So was it grow on plates, and have you tried anything related to moving DNA by either plasmid or I just wanna kind of see how the cell will behave with obviously genome transplantation or just plasmid transfer in this new media.
Thank you very much. We tested the agar plate, and we saw the Friday colony in the new media, serum-free and the defined media, but we didn't test it the outside the DNA. So I don't have any idea about that. So
Okay, but that's great on the agar media. Yeah, that's amazing work. Really exciting. Thank you.
thank you very much.